Thank you so much for joining us today, for showing up after first Hospitality Suite evening. I really hope you enjoyed and had fun. And today we will have some fun with breakfast seminar and launch of Spectronaut 21. So let me introduce agenda for today. First, I will give you a short introduction to Biognosys Group, and afterwards we will have a first speaker for today. Monika Pepelnjak will talk about what we prepared for Spectronaut 21, what are the updates, what are the improvements, and afterwards we will have a first external speaker, Pouya Faridi from Monash University, talking about the directDIA for soluble HLA immunopeptidomics and how that leads to identification of the disease-related peptides in plasma. Afterwards, we will have another external speaker, Ben Anderson from University of Wisconsin-Madison, talking about mouse multi-PTM atlas. So let me start with the introduction to Biognosys Group. Biognosys Group unites Proteomics, Biognosys, and Biocrates. Within this organization, we can offer comprehensive solutions for the proteomics, metabolomics, and lipidomics. While being vendor neutral, we can offer solutions for the sample preparations, separation of the peptides, and data analysis for the major platforms. And besides that, we also offer CRO services for proteomics and metabolomics. And with that, I would like to give a stage to our first speaker. In case you will have questions, please hold them till the end of all the talks, because we will have a common Q&A at the end. Monika, welcome.
Thank you, Monika. So, good morning, everyone. It's my great pleasure to introduce to you what is new in Spectronaut 21. So, to start with, we wanted to first look back at Spectronaut 20, where we introduced the novel module Kuiper search engine that significantly improved the speed and performance for immunopeptidomic searches as well as PTM probing. So now for Spectronaut 21, we went back to our basic modules and see how we can improve, and we asked 3 specific questions. One is whether we can improve the core machine learning concepts to boost our protein identifications. Then we looked into our AI models and specifically addressed the question about unseen PTMs. And we also focused on the performance, specifically increasing the performance on cloud. So I will focus on AI models and the unseen PTMs. So what do we mean by that? Our training or our models are trained on unmodified and modified peptides, and they cover 23 different PTMs. However, we allow to search you with different PTMs as well, and those would be characterized as unseen PTMs because the model hasn't seen them before. So the idea is that you have, because the models have seen some PTMs, it will perform well for those. However, for the unseen PTMs, you can expect worse performance. This can be seen in a plot like this. So basically, if we are trying to predict our retention time, you can see that for seen modifications or unmodified peptides, we are quite good at predicting the retention time. However, this is not the case for the unseen modifications. So how do you address this challenge? The first thing is that you should encode your modifications well. We have been working on this for a while because it's quite a challenging thing to do, because you want to encode the modifications in a way that they will perform well for the unseen PTMs. However, you should not lose your performance on unmodified peptides or on the seen PTMs. So we have tested different types of encodings, such as token encoding, where each PTM is presented by a token, atomic encoding, where you basically write out your PTM based on the atoms that are in there. Then gain-loss encoding, where you can you you encode it based on what was gained or lost when the modification was added to the peptide. Or you can also try SMILES encoding, where you basically write out or encode the structure as well. If you want to learn more about how these different encodings work, I would suggest you visit the poster from Alex on Wednesday, sorry. So, bit of spoiler alert, we then went with the atomic encoding. Why is that? Because we saw really good performance in the improvements in the prediction. I'm showing here improvements in prediction for 3 different unseen PTMs or labels between Spectronaut 20 and Spectronaut 21. So you can see our absolute error for the predictions went significantly down for all 3 unseen modifications or labels. We also tested that we did not lose the performance on the unmodified peptides and seen PTMs. So you can see here the performance with the atomic encoding for tryptic and unspecific peptides. And you can see that we are actually performing better for those predictions as well. So I showed you that we have improved our models, but what does this mean for the end users? So did we actually improve in anything afterwards? So we have seen by combining our improvements in the machine learning scoring and our new AI models, we have around 10% improvement in protein identifications on average, and this was done by benchmarking 100 data sets. You can see, so each dot is a data set. You can see that we have some outliers that perform much better, but on average, we are around 10% and median a bit less. So we have seen these improvements, but we wanted to see also that our FDR is well controlled. So we do entrapment analysis to test that our FDR is well controlled. You can see that we do this on global protein level as well as global precursor level tests. And we could see that our FDR was well controlled, and you can see that it is controlled as well for single cells, while the entrapment FDR is a bit more difficult to estimate because of the higher variability. So we have seen these overall performances, but we wanted to see as well whether with the improvements that we have gained, you would get any additional biology as well. So to test this, we looked into our P2 enrichment, so our P2 plasma data sets. And for those 11 data sets that we have from different instruments and different proteins, we saw similar improvements, around 7%. And what was interesting for us, or one might even expect, is that the abundance range of the newly identified proteins is significantly lower than the average abundance of the proteins. So we are really starting to identify these low abundant proteins. While you can see here, it's a very small distribution of the lost proteins. So those would be just randomly proteins that we randomly missed from Spectronaut 20. This is an example for one P2 data set. We have seen this across the 11 data sets that we have tested. So it's always the case that we are now identifying more low abundant proteins. We also looked where do those proteins come from? Is this something biologically relevant, or are we just picking up more noise? We have seen that we actually saw enrichment for transcription regulation and DNA binding, and this is again example of one P2 data set, but when you summarize the terms, we saw actually that we have chromatin binding and transcription regulation activity across the 11 data sets. And so basically, in biologically relevant samples, we could identify more low abundant proteins that are enriched in transcription regulation activity and chromatin binding. So this was one case study. We also, since I said that we also improved in our models for modifications, we also looked into the phosphorylation analysis. For this, we looked at ground truth phosphorylation data set, where 225 ground truth phosphopeptides were spiked into the yeast background. And we could see that With Spectronaut 21, we have more identifications of true, so we look here at total localized precursors. So you count identification, the same ID across different runs more than once. So that's why we have the numbers that are much higher here. Just want to point this out. But we saw that we basically have good or better identifications with good localization rates. And what is more, and goes in line with our identifying more low abundant proteins as well, we saw specifically better recovery for the low abundant, so when a small amount of peptides were spiked in. We then looked at another case study for phosphorylation. This is EGF treatment analysis where the cells were treated with EGF, and what we could see is that we have improved the data completeness for localized peptides. This meant that we had more localized sites for the differential abundance analysis and also more phosphoproteins that we could profile and then do the differential abundance on. And this also meant that we then identified more significantly different differentially abundant proteins. We did the geoenrichment, pathway enrichment then as well, and you can see that we are actually hitting the same terms with both, so Spectronaut 20 and 21, so that's a good sign, but we saw a higher enrichment in Spectronaut 21, so we seem to be picking up relevant biology here as well. The next thing we looked into more specifically is the immunopeptidomics. As I said, between Spectronaut 19 and Spectronaut 20, we had massive gains in identification and speed improvements, and we improved that a bit more in Spectronaut 21. So you can see we have even higher identification and even a lower processing time. And again, this is done so we run, each dot is an experiment of immunopeptidomics that we have done. So we see this on broad number of data sets. What is more, we also looked into this specific experiment with the JY cells. You can see that between Spectronaut 20 and 21, we have more identifications, while the fraction of the peptides that are considered non-binders, so those gray ones here, basically stays constant, so we are not identifying more non-binders. What is more, we added with Spectronaut 20.4 already the detection for in-source fragmentation. So basically, it would flag the peptides that have, so basically the parent and the child peptide that are of the same in the same sequence ladder that elute at the same time. And we have seen that with this, we can improve our data quality further. So from this experiment, you can see that if we look at the predicted binders, we have almost no in-source fragmentation, while for the non-binders, there is a significant fraction of in-source fragmented peptides. And you can now, with Spectronaut, directly filter them out, and then with this, increase your data quality. If you want to learn more about this, Monika will present a poster about it on Thursday. So I talked about analytical performance now so that we can recover more relevant biology, but we also improved the computational performance. So as you might have heard already, you can run Spectronaut searches in parallel. With Spectronaut 20.3, we have introduced a new additional step in the workflow on how you should parallelize your searches. So you basically you can parallelize the so-called pulsar step 1, and then you have a merging step. You do then pulsar step 3, and then you do a merging step, and then you do your DIA analysis in a parallel way as well, and then you do the S&E merge at the end. With this introduction of additional pulsar merging step, we have significantly improved the reproducibility or consistency between running the experiment all at once versus running it one sample at a time, so in parallel way. Omics, we have a solution for this that will orchestrate your jobs in parallel. So it's called Omics Flow. But we have seen, and we're very happy to see that others have implemented their custom solutions for this as well. So we are very happy to see this used by the others as well. But we know that not everyone can run on cloud. Oh, no, sorry. We also saw that the performance, so we improved the performance for Spectronaut if you look at the performance on Omics Flow, you can see between Spectronaut 20 and Spectronaut 21 that we have reduced the time required for the pulsar step 1. So those are the parallelized steps. And more importantly, we improved the time needed for the merging steps. And this is important because those are usually done on the more expensive nodes. So this is the cost-saving improvement as well. And you can learn more about this at the poster that will be presented on Wednesday by Tejas. Now, yes, not everyone can run their things on cloud. And we have seen that you can save your time as well by basically just subsampling the runs that go into the library generation. And now we have added an additional setting that you can use that will subsample your runs that will go into the library, so your library generation step will be much faster. And how this affects your final results is also included in the poster that Tejas will present on Wednesday. So if you're interested in this, stop by. So with this, I would already like to conclude. So Spectronaut 21 is available today. And we have shown, I showed you that we have around 10% more protein groups on average and 11% more on immunopeptides. We have improved our computational performance. We have improved our deep learning models. We also have, I did not talk much about this, but we also have improved the user experience. We have improved our PTM analysis, and we have done substantial benchmarking to ensure that our data quality is high. If you have any feedback on what you would want to see in Spectronaut 22, we would be very grateful if you would write to us or contact us, talk to us at a conference. You can also always find us at the booth, and you can visit our Spectronaut 21 launch page here. So with this, I would like to thank you, and you introduce the next speaker. Thanks.
Yeah. Thank you, Monika. And now I would like to invite our second speaker, Pouya Faridi.
Thank you. Don't need that one. Cool. Thanks for the invitation and opportunity. Okay, today I'm going to talk a bit about what we are doing in the lab in terms of identification of peptide HLA complexes in body fluids. Okay. So I think this is now a concept that everyone knows, that we are trying to find biomarkers for diseases in body fluids. And it's clear why, because it's difficult to get always access to the tumor in the body. We have always limited material. And when you want to monitor the disease across the term of the treatment, it's not easy to get biopsy like monthly. And now the idea of liquid biopsy, it could be ctDNA, it could be plasma proteomics, is something that there are quite a few groups in the world are trying to use them to understand what's happening in the body, especially for cancer and tumors, because it's non-invasive. It's something that enables us that during the therapy, during the treatment, understand what's happening for the tumor. So this is something that people are using, but we want to use it in another way, and we want to use another compartment in the body fluids, which are peptide HLA complexes, which are in the plasma or other body fluids. There were quite a few presentations in the last few days about what is peptide HLA complexes. Just briefly, all the intracellular proteins get degraded by proteasome, then the small peptides bind to a molecule called HLA. And the majority of HLA peptide complexes come on the cell surface. But there are a few, around 1% of peptide HLA complexes that can release to body fluids with different mechanism. And also when we have cell lysis, we get also peptide HLA complexes released from the tumor or even like the normal cells and go to the body fluids like blood, plasma, CSF, and urine. So there are several studies that, yes, the complexes stay intact in the body fluids. And why it's important that we can look at the peptide HLA complexes. I mean, the best technologies that now we have, we can only look at like around 10,000 proteins in the plasma. And some of the intracellular proteins never make it to the plasma. So these are always remain there in the cell, or they get degraded by different mechanisms in the cell. But when we look at the immunopeptides, we have 2,000 proteins. But immunopeptidomics tell us what's happening actually in the cell. So all the peptides that we are getting somehow showing as a marker of intracellular protein. Also, I think you heard in the last few months about microproteins. We have short proteins with very short lifetime. They have roles in the cell physiology and also pathology, which is very difficult to find them, and we don't know if they are stable enough that we can detect them in the body fluids. But immunopeptidomics can show them, can preserve them in this context. But what is the challenge? So we know that peptide HLA complexes are there for a long time, but one of the challenges is always sensitivity. So they are diluted peptide HLA complexes in the body fluids. And one of the problems was that if we want to look at the plasma immunopeptidomics, we need to have like a few milliliters of the plasma sample, which is in a clinical setting, it's not something easy to get access to. The other challenge was throughput. So we're talking about biomarkers. So usually we need to have enough number of samples, we need to have a robust and like high throughput method. And immunopeptidomics by nature is a laborious work. It has multiple steps for sample preparation. So we need to have an automated method to do this analysis on several samples. And reproducibility. Usually when we have a step, when we have a procedure which is multi-step and we need to do it by hand, then the reproducibility decreases, and then we cannot compare data that we get across several patients at the same time. So what we did, we tried to have a workflow, which we call it subroom 2. So we have subroom for normal HLA class 1, which is for high throughput workflow for soluble HLA immunopeptidomics. So we design all our immunoprecipitation on the instrument Kingfisher, so we can do sample preparation for 12 to 96 samples per day. We use magnetic beads, which are protein A, magnetic beads, which are covalently bind to antibody that we use. Here it's W632. And at the time that we started this procedure, so we thought, okay, we need to do DIA, but it was not possible to do direct DIA, or at least we didn't have the technology there. So we had to generate a spectral library and then do the DIA search based on that spectral library. So we developed this technique, and the first thing that we wanted to see was that, okay, how low we can go for soluble HLA immunopeptidomics. We made a serial dilution from 100 microliter to 1 ml of sample from the same donor. And as you see, as expected, we have more input material. We can get more peptides from like 1,500 to 4,000 peptides from 200 to 1 ml of plasma. All the quality control, as we expected, passed because like the 9 mL of the main one, more than 97% of peptides that we identified were binder based on the prediction algorithm. And then there was a very clear similarity on the HLAs that these peptides can be bind to. And what we wanted to know also is that if we go for like higher amount of material, we can capture the peptides we see in the lower input. And this was the case. So the majority of peptides, which were in 100 microliter, input you can find in the higher volume, and as you see, etc. like this. So we have a core immunopeptidome that when we increase the input material, we can see more peptides, but still that core immunopeptidome remain there. So it's not only about identification. We wanted to know if we can also have some idea about quantification. So what we did, we looked at the intensity of these peptides and also to see if we have a difference in terms of the abundance, can we get this information in the soluble HLA? And you see this very nice trend for the same peptides that we see across different samples. By increasing the input material, we see this increase the intensity there. The other question that we wanted to answer, so if we do the same experiment on the same sample in 2 different days, if we get similar results, so it's like intraday like stability, and also samples from different patients. Of course, we have different amount of soluble HLA in different patients, but we find a very good stability across samples that we analyze in different days. And you see that this was not only, again, about the number of peptides, also the intensity or the abundance of these peptides. Okay, so we have the workflow, and now we want to answer some biological questions. The cancer that we focused was melanoma. This is the model cancer in immunotherapy for cancer vaccine for immune checkpoint inhibitor therapy. So, but there is no information about the soluble HLA in melanoma, and it's quite dynamic. So across the treatment, lots of patients go through neoadjuvant immunotherapy. The immunopeptidome could change, and we need to understand at which time which peptides are presenting actually on the tumor. We have a very good understanding on the cancer antigens in melanoma, and indeed, we are part of a big clinical trial on cancer vaccine combination with neoadjuvant immunotherapy, and we wanted to make sure we can use soluble HLA there. So what we did, we received 5 samples from 5 patients that exactly at the time of the tumor resection, we get also the plasma samples. So at the same time, 2 different compartments, 1 plasma, 1 tumor. And that was good because technology improved on 2 different ways. So one was on the instrument, because we could do very narrow window DIA, fast instrument. On the other part, we could do directDIA data analysis. So we have both instrumentation, and we have also the software which can analyze this data. So we have this technology now in hand, so we don't need to generate spectral library. So we improved our workflow to directDIA, and we use Spectronaut for our data analysis. The other new thing that came was the good database from non-coding, non-canonical off-derived peptides or cryptic antigens. So we also put that one in our search strategy, and we analyzed our samples from 5 patients. The first thing that we saw, it's a clinical setting, so you always not get the same amount of material from different patients. So sometimes you get around 200 microliter of plasma, sometimes you get more, and then the amount of tissue that you can get would be different, less than 10 milligram to up to 30 milligram. So we did immunopeptidomics, and what was interesting for us is that it's not always input material that tell us if we see more peptide or less peptide. We find there is a correlation between the number of peptides that we can identify based on the patient. So for patients that we had, if we divide the number of peptides that we find to the volume of the plasma or to the milligram of the tissue that we get, we find this nice correlation across all the samples. So we introduced these new indices in the field that we call it like tissue recovery index and plasma recovery index. And we find this this is at least in 5 patients, so we need to expand it more. This is something happening which is similar between plasma and tissue. So these are the number of peptides and the protein sources that we found. As expected, we see usually more peptides in the tissue than plasma, but still we are getting a good number of plasma immunopeptidome for 5,000 to 10,000 peptides. This patient 4, was quite interesting. We find more peptides in plasma than what we find in tissue. quality control for immunopeptidomics. You always see this slide everywhere having peptidomics. You want to make sure that 9-mer amino acids are the main ones, which is what we expected to see. The first thing that we wanted to do after this quality control is that are there particular HLA types which are preserved more in plasma than the other ones? So what we did, we used net MHC prediction to assign each of the peptides to all the HLAs which the patient has and see what proportion can bind, what peptide can bind to what HLA. So there are cases that we see the peptide can bind to more than one HLA, and it's why you see the percentages are more than 100% if you add them up. In most cases, we had a very similar trend for what we see in plasma and what we see in tissue. But what was interesting for us was HLA-B8. So in HLA-B8, you see we have like half of the peptides, like if you think about the percentage, that we have in plasma when we compare it in tissue. So it was good that we have B8 in 4 different patients. So this patient, B8 is like this. This patient is 16% to 30%. This patient is from 20% to 55%. For patient 4, it was different. So we have 29% B8 peptides, 25% B8 peptides in tissue. But what we did, we just reanalyzed data and we assigned peptides to the best binder HLA. And when we did this for patient 4, we found that we are going from only 5% B8 to 15%. So looks like that there are some physicochemical properties for some of the peptide HLA complex that can keep them more stable in plasma than the others. The next question we want to answer if we are seeing same peptide there or if we are seeing the same protein sources. What we found across all samples, around 30 to 50% of peptides that we can see in plasma samples, in tissue samples, can also be seen in plasma. But when we look at the source proteins, we have a more shared source proteins between plasma and tissue. So that tells us that maybe some of the peptides stay more on the tissue, some of them stay more in the plasma. But this is what I come back to this a bit later. In terms of intensity, we expected that plasma is kind of a diluted version of the peptide HLA complexes that we get from the tissue, and this is exactly the case for all the peptides that we saw that's shared between plasma and tissue in all patients. We see a completely significant difference in terms of the intensity of the peptides when we compare it between plasma and tissue. Cryptic antigens, so these are the peptides that are present from non-canonical open reading frames. So this is a hot topic, especially for immunopeptidomics these days, because some of them could be cancer specific. So when we look at the presence of cryptic peptides in plasma and tissue, we see like maximum 1% of the whole immunopeptidome can be cryptic, which is in the range that we expected. We see a little bit less in the plasma, which is again what we expected, so it's diluted. Again, 9-mer are the main ones. What was interesting for us is that it's around 40% of the cryptic peptides that we see in the tissue are actually presenting in the plasma. So plasma can preserve these cryptic peptides, even we know they have a lower abundance than like normal canonical peptides, still we can see them. And what was also very interesting for us, there are some of these cryptic peptides that are shared between different patients. So these are the cryptic peptides that we found shared at least between 4 different patients out of 5 patients and present at least in 1 plasma sample. Based on all the in-house study that we had on healthy tissues, these has never been reported in any healthy tissue, and this could be melanoma specific or melanoma associated cryptic peptides that can find on tissue and also plasma. Melanoma, very well known antigens. We have melanoma associated antigens, very well established cancer associated antigens. We look at these known antigens in plasma and tissue. We see around 10 to 12% of the peptides that we find across plasma and tissue are coming from these sources. Again, we look at the intensity of these peptides to see if the abundance is similar or different in plasma or tissue. In most cases, again, we see that the cancer-specific antigens are diluted in plasma, but still detectable. But when we look at this very well-known list of cancer-associated antigens in melanoma, we found that some of them are quite interesting. And this one I want to show here is like PRAME20. So PRAME is a very well-established, PRAME20 very well-established melanoma-associated antigens clinical trial across other versions of PRAME. And what was interesting, we see more peptides from PRAME20 in plasma than tissue. So there are antigens that we may not see them on tissue, or for like less abundance there, but we can see them in plasma as biomarkers. So what we did, we have melanoma samples and we have all the sources that we found across this patient. We have in-house data and also some published data about healthy plasma. We have HLA ligand atlas, which is our reference for all the peptides that present on benign tissue. And we have a data set from the peptide HLA complexes in plasma samples from autoimmune disease. So we compared the source antigens across all of these 4 samples. And we found that around 247 source antigens are uniquely present in melanoma samples. Some of them are unique to plasma, around 80, and 167 is shared between plasma and tissue. And we found 9 source proteins, which I identified in 4 out of 5 patient samples, which are these peptides. These are these source antigens. So we find 9 antigens only in plasma. We can only in plasma, only in melanoma. And if you look at them, some of them has never been reported in IED, which is our atlas for all identified immunopeptidomics data sets around last 20 years. So we put all of them together and we introduced them as potential melanoma-associated biomarkers, which at least so far we don't have any evidence of their presence in plasma samples from healthy tissues. And now we are expanding this to a bigger cohort from 5 to around 20 patients to see if we can validate them in more patients or not. By this, I hope I convinced you that now we are in the time that we can do immunopeptidomics on soluble HLA in scale. Plasma immunopeptidomics presents some sources which are unique to soluble HLA, and some of the peptides are uniquely presenting there. Circulating HLA in plasma capture some of the tumor-derived and also cryptic peptides, which are tumor-specific. And I think in future, we try to use this information in some of the clinical trials that we have for biomarker discovery and immune monitoring. So thanks to my team, especially Erwin and Gabe, who are leading this soluble HLA project in the lab. All patients who agreed that we can use their sample, funding body, and thank you.
Thank you so much, Pouya. Great presentation. And I would like to invite our last speaker of today, Ben Anderson. Please.
Okay, can you hear me? Microphone, sounds good? Okay. All right, thank you everyone, good morning. I'm gonna go over a lot of the content that I went over in my slides yesterday at my talk. If you were there, thank you for coming, but I'm just gonna give a quick rehash. But stay towards the end, I'm gonna give you some tips maybe for working with PTM analyses in Spectronaut, and then also talk about some exciting new results with the PTM probing mode. Okay, so we all know that PTMs are important for biology, but there's an unsolved problem in PTM analysis, and that's we have no genome for where PTMs live. We're not lucky like proteomics, where we can have a full end-to-end knowledge of where all of the proteins are and what they look like. So every time we get access to new and better mass spectrometry, we should probably go back and look into what we can see in our mouse or any tissues to see what kind of depth we can see for PTMs. And when you have this deep mapping of all the PTM sites, you can get a ground truth, which is really useful for a lot of different analysts out there. Like, for example, you can give it to the computational biologist who can use this to train new models to predict where future PTM sites might be. Okay, so a couple years ago, we had access to new high scan speed, very sensitive instrumentation. This is the Orbitrap Astral, and to see how well we could do with this, we attempted to make a PTM atlas of phospho in various mouse tissues as listed here. We published this back in 2024. And with just 48 mass spec injections over about 20 hours of acquisition time, we got 81,000 sites. So, and honestly, part of the reason why we also did this was because we had access to really good software as well. So the main story here was probably the instrument, but honestly, in the back end, having good software was really important as well, and that was Spectronaut for us. Okay, but we look at this as saying, we're not at the top of the mountain. We're trying to climb this PTM mountain to have full knowledge of it, but honestly, we can't even see the top of the mountain up here. We like to think that basically we just set a flag down at Basecamp Phospho, and we need to figure out the rest of the way. And to that end, we have the Coon Lab Mouse Tissue Pan-PTM Atlas project, a multi-year effort to catalog all the mouse tissue PTMs. And so far, we have these 4 PTMs done so far: phospho, N-glyco, acetyl, and ubiquitin. So I'm just going to go over some results from them really quick. Methods first. So basically, for all the atlases, is the laser working? I don't think so. That's okay. So for all the atlases, we have 12 to 15 tissues per atlas. We have a subset of about 11 that are found across all the atlases. All of them underwent some amount of non-enriched proteomics. So we pulled them out in order to see, use them for input normalization. They went through these enrichment methods. All of them went through astral DIA or astral zoom DIA. And then we have various fractionations. And for these, the phospho, acetyl, and ubiquitin were Spectronaut directDIA analysis. And then N-glyco is through FragPipe. Maybe someday Spectronaut can do glyco, but we'll see. And this is the total acquisition time. And here's the analysts up top. So phosphoatlas at a glance. This is basically the same plot as before. However, this 82,000 number is after reprocessing with the most recent version of Spectronaut, version 21. So we got about 1,000 more sites. Here's our U-shaped distribution in the bottom left, showing the number of tissues that each phospho site was detected in. So we see most of the sites are detected in just one. A largest number in terms of but then you see kind of a increase towards the end. Here's the places where they localize within the cell. We integrated some other databases like AlphaFold and showed that phosphosites tend to aggregate towards more disordered regions of the proteins. And we did this localization probability test where we looked for phosphoserine, threonine, and tyrosine and found that they had higher localization probabilities with better confidence, whereas phosphoproline, which is not a thing at all, and that's our false discovery estimate, is more towards the right. And then Monika in the first slide actually showed another test that we did, which was using the spike in phosphopeptides. So that was 2 checks that we did to ensure that we could trust the data coming out of our instruments and with the DIA method. Here's the acetyl atlas, so 22,000 sites, again, a U-shaped curve, and here's all the places where they fall. Oh, yeah. Oh, okay, thank you. Oh, I have to double, okay, yeah. So this is our, so acetylation is really important in ribosomal activity. This is a set of 360 acetyl sites that we detect in the mouse ribosome across all these tissues. This is unprecedented depth for acetylation in mouse ribosomes. And then I worked with a really talented biologist who helped me do some analysis to show that this is important for active sites on the ribosome. Okay, ubiquitin atlas, 95,000 sites. We were very impressed with that. Again, a U-shaped curve. This is the distribution across the cell. And we found that if you actually increase the loading amount, you can get more ubiquitin sites without loss of any peptide specificity during your enrichment. And this is an interesting story. So some of you may know that in order to validate acetyl lysine sites, you can use a diagnostic ion. I think it's about 114 MZ, if I remember right. And basically, it's the lysine plus an acetyl, and it forms 2 different kinds of ions. It's like 114 plus the 17. But we can also do something similar for ubiquitin sites. So instead of having an acetyl here, we can use the gly-gly remnant, which is diagnostic for ubiquitin sites, and then we can get these 2 masses. And then when you go back in and look at the precursor apex, which is a column you can get from Spectronaut, and you go back into the raw file and say, do we see 215 or 198 with some ppm mass error tolerance, then at least at the precursor apex, about 80% of sites have at least one of those 2 ions. And then if you look at the XIC, we're actually seeing really good patterns of rise and fall for these ions as well. So this is indicating that this diagnostic ion also works for other acyl lysine modifications. And if you want more details, go talk to Riley at Wednesday, poster 612. Okay, N-glyco, like I said, N-glyco is not done with frag pipe. You'll notice that this U-shaped curve is lower down. Is this a fragment of the enrichment? Is this just more difficulties with N-glyco? Or is this because the software is frag pipe? I don't know. We'll have to see. And then you can get various different types of N-glycans. Okay, so in total, we saw 15,700 proteins with NEPTMs. And this is not inflating with isoforms or Trimble proteins. This is just solid one sequence per gene proteins. We have 196,000 sites, 213,000 sites plus PTMs, which if you don't understand what the difference or how that comes about, it's because we have a bunch of glycans causing different PTMs to live at that one glycosite, but then we also have 12,000 lysines with both an acetyl and a ubiquitin detected at some point in our enrichments. And in total, Spectronaut, I should say, the Spectronaut plus frag pipe found 750,000 unique LC-MS precursors. So on average, we have, what is that, like 2.5, almost, or over 3 different precursors per site as evidence. So it's not just a bunch of one-hit wonders. Okay, in total, 125,000 sites is the best in lung tissue. These are the 11 tissues that we found in all the that we used for all the atlases. And this is a subset of additional tissues that we had in just a subset of the atlases. So we have these 4 white adipose tissues, and then in acetyl and ubiquitin, we added female, and we ended up getting ovary on those as well. Okay, number of sites per protein. So yeah, the highest number is just 1 site per protein. However, these are mostly low abundance proteins where it's probably hard to see just any peptides from them, so getting you 1 site was impressive. But then we also have this large distribution towards the right side where we have a lot of proteins with a lot of sites. And towards the very end, if you're curious, the protein with the most sites was Titin, which has like 500 or 600 sites, and most of them are phospho. Okay, just to break down, so this is proteins with just a single PTM. Phospho led the way with 2,000, and this is any protein that has 1 or more phospho and nothing else, right? And if you look at the 2-way subsets, this is dominated by phospho plus ubiquitin, almost over 3,000, and this is because of, well, probably because we have a lot of phospho and a lot of ubiquitin sites detected in our atlases, but also because they tend to coexist for regulating cell degradation, and that's a known thing from biology. And also, interestingly, we have about 1,000 proteins with all 4 PTMs detected on them. Okay, so like I said, we did total proteomics non-enriched from all 4 of our atlases. When you combine them together, we found 16,000 proteins detected in those non-enriched samples. 1,900 ended up not having any PTM detected on them after doing the enrichments. And if you look at how these PTMs break down, so acetyl tends to be more towards the more abundant proteins. Phospho and ubiquitin, a little bit kind of the same trend, but not quite. And then N-glyco is kind of distributed across the abundance range for all these proteins. And if you take those 16,000 proteins, put them on the left side of this Venn diagram, we have the 1,900 that have no PTM. And then the right side of the Venn diagram is the 15,600 proteins that we detect with any PTM site that's on the table before. And if you think about that, that means that we have a bunch of proteins, 1,300, that were not detected in the non-enriched. So this means that we were able to pull out of the weeds some evidence for some proteins that we just never saw with our really deep DIA total proteomics. And this is dominated by phospho, probably because phospho is like more abundant, higher stoichiometry, I should say, but also that titanium IMAX seems to have really good ability to pull things out of the weeds, it seems like. At least that's my theory. Okay, so we compared it to these 4 databases, and we pulled out just the sites that are unique to only this atlas. To be fair, we did not look at the overlap between all 4 of these other ones. That would make the Venn diagram too complicated. But if you just look at what we found that nobody else found, that's about 80,000 sites. Okay, so if you're thinking, well, you found 80,000 additional sites that no other database, or at least these databases, haven't annotated, that just means you're pulling things out of noise, and it's probably just a bunch of junk. Well, I went and looked at that. So this is a comparison of the 80,000 sites that were only in this atlas, and then 120,000 sites that we detect in this atlas, but were also annotated in one of those 4 databases. And we show that for our 4 PTMs, the PTMs unique to this atlas have about 2x lower abundance compared to sites also found in the other databases. And if you look at like phospho, that's particularly striking. I think it's like 3x lower on average. So, okay, and of these 80,000 sites, so this is the distribution of the tissues they were detected in. So this is the 11 tissues in all atlas, and then I aggregate all the white adiposes together. So about 50,000 of them were found in more than one tissue, which means, again, multiple sources of evidence, which kind of boosts the claim that it's not just a one-hit wonder. And if you look at these 30,000 single tissue sites that are new, 83% of them have multiple precursors or were found in multiple replicates of that tissue. So this is further evidence. And I guess you could say at the end of the day, only 5,000 sites, so 17% of this bar are just one-hit wonders where you have a single precursor in a single sample. And this is 5,000 out of our total of 120,000 sites. So, or I should say, just for the, yeah, so it's about 8% of the sites that are unique just as Atlas are just one-hit wonders. Okay, and then if you look at the sites or tissues with a new PTM site that's only found in that one tissue, so this is the same 30,000 as over here on the left. We have brain and liver up near the top, so you'd think that with these really commonly studied tissues, they'd be less likely to have newly found PTM sites, but this goes to show that if you go really deep and you have good instrumentation and good software, then it's very possible to find additional sites even in these well-studied tissues. We also had really good coverage across histones. I'll go skip over that. We mapped them to SNP variants, and we looked at the SNP variants specific to PTM sites. So we mapped this to the ClinVar database, which is a database of pathogenic clinical variants in human. So we did some human mouse homology, and then we showed that there are about 800 total sites with pathogenicity at the PTM site across all of our PTMs. And importantly, when you have a SNP at a PTM site, that's about 3x enriched compared to the background of SNPs at non-PTM sites. And we use this during our Acetyl Atlas to show that the SNP here, the SNP that exists at this new acetyl site was probably key for causing the active site binding of this metal ion for this disease. Okay, let's talk about some Spectronaut things. So I think it's always important whenever you're talking about software to give people a sense of what kind of parameters they've used. I've done software development in the past, so bear with me. So for our Atlas, it was really important to turn off cross-run normalization. A lot of people leave this on and don't really think about it, but when you have really, really different, disparate sample matrices, it's important to turn it off. So if you have like really similar things, like it's a bunch of plasma, it's probably safe to turn it on, to leave it on, but when you have, brain versus heart versus lung, whatever, that's really, really different. We got much better results, more stable results, and more sensible results with the biology when we turned off cross-run normalization. And importantly, input normalization strategy. So this is something that I had to go find from the Biognosys YouTube channel. So when you're in the setting up the experimental parameters in Spectronaut, you have the choice of setting the input normalization and the stoichiometry calculation strategies. So input normalization, you have to understand which parameter to use. There's 3 options. You have all peptides, non-mod peptides, and non-modifiable peptides. So when you have high stoichiometry PTMs, you should use all peptides. And then when you have low stoic, but the PTM is on a common residue like lysine, for example, acetylene ubiquitin, use non-modified. And if you have something really uncommon, like you're looking at, I don't know, tyrosine oxidation or something like that, you would use a non-modifiable peptides. Or you have highly variable stoichiometry between samples. Maybe you have like one that's depleted and one that's super enriched in some way. And then remember, if you do PTMs, try and collect some kind of non-enriched total proteomics so you can benefit from the stoichiometry, and you should always use your non-enriched total proteomics as input, or ideally if you can. Okay, so some other parameters to think about. For the default peptide length, this is between 7 and 52 residues, but for acetylene ubiquitin, because they live on lysines, they do not allow trypsin to cut there. It causes a miscleavage naturally. So the peptides are naturally longer. So you might want to bump this up if you're doing PTM analyses with acetyl or ubiquitin, or any other acyl lysine. Site localization, so 0.75 is a generally accepted minimum. You probably shouldn't go lower. You might want to consider going higher if you want to control your false discovery a little bit, but definitely you can play with this after the fact when you go and filter your PTM site report. PTM consolidation, yeah, so sum versus linear model. Sum, I don't know, I'm still torn about which one to use. The sum is the default, it's pretty good, but I can see cases for using linear model, so I encourage you guys to read the manual and get informed on that one. But linear model might be better for more sparser cases when you have, but also you have lots of different precursors that you have very different ionization efficiencies and therefore would cause weird artifacts in the data just because of the sum modeling. And also PTM multiplicity. So if you leave it on, then you're separating peptides that have multiple of the same PTM on them and they become their own row in the PTM site report. But if you turn it off, then you're just combining it and then you're assuming that all the quants that you're summing up together just are the live on that one site, but you're going to get duplication of the summing events when you actually go to do that. So again, read up on the manual on that one, and it's important to understand what to do with that. And I'm tempted these days to leave it checked on so that I can understand the proteoform level information that comes with the multiplicity of the PTMs on various peptides. Okay, so using the PTM site report, a journey through the 5 stages of grief. So I'm sure many of you have heard this before. There's like 5 stages of grief: denial, anger, blah, blah, and acceptance at the end. A couple of people in the lab have gone through this process before. We look at the PTM site report, we get kind of confused, and we're like, oh, this is not what I need. And then we go through and we say, oh, we need to like change it to be something else that by various manipulations or whatever. And we go round and round and round in a circle for a long time. And then eventually we're like, wait a second, no, this is exactly what I need. So I encourage you, if you think that the PTM site report is not for you, don't try and do your aggregation beforehand on the PTM site report. Take a step back, let the PTM site report be what it is, just a long form quants, and do your aggregation in other tables. So for example, I tried to make the PTM site report be the list of 213,000 site plus PTMs that I showed in that previous table, but that was doing too much. I wanted to keep it separate based on the protein source because, like for example, I wanted to be able to separate the confident non-Tremble proteins from the Tremble proteins. So in order to do that, I had to keep the PTM site report as is and just make a separate summed quant table and then a separate site report table, right? And if you have any questions about how to manipulate the PTM site report, please send me an email. I'm happy to give you all sorts of information about that and give you scripts and whatnot. Okay, so yeah, like I said, there was an initial problem. How do you aggregate across isoforms and stuff like that? And the answer is you just don't do it. You aggregate them as needed for your quant or your homology later down the line. Okay, and yeah, so this is the PTM site report. If you're not familiar, you export it from here. This is the report tab. This is the one that's typically available when you do total proteomics. This is the standard report. And this is grayed out when you do standard proteomics. But when you actually go to do a PTM analysis, this becomes ungrayed. And then you have the option to export your protein group and PTM columns only. PTM columns are new when you do the PTM site report. There is no peptide, elution group, or fragment columns because they've already done the hard work of aggregating all that for you in this final report. So my tip is just export the PG dot quants, the PG dot protein groups, and then just all the PTM site report data, all these new columns. And finally, PTM probing mode. So there's this new development from Spectronaut 20. It's the Kuiper engine. So basically, when you do regular PTM search, you have fixed mods and you have variable mods, and then, for example, you'd add ubiquitin. This is what everyone tends to do for their PTM. However, there's a new mode called PTM probing. You, again, have fixed mods. There's known variable mods. So I know for a fact that most of my peptides will have one of these 3. But then you probe for an additional bunch of variable mods. There's like 30 plus. So you can do other cool things like look for a cell or phospho, and they have a whole bunch of other things. They even have like sodium adducts, which makes me feel like I'm doing metabolomics again. And I was curious because I was thinking that ubiquitin would also, when you do the ubiquitin enrichment, it would enrich for other acyl K PTMs on my peptides. So I was like, okay, let's give it a shot. So like I said before, 95,000 ubiquitin sites from the single PTM search. That's the number I showed in the data before. But then when I turned on PTM probing mode, I found 94,000, so basically very little loss. But then I also found a bunch of additional really cool sites. So dimethyl, trimethyl, crotonyl, which is interesting for a lot of reasons. I found 2,000 phosphosites. I found 1,300 acetyl sites, 800 succinyl sites, and all sorts of cool stuff. And I was like, okay, well, maybe something's different about them. And I looked at the quant for these gly sites. And for the overlapping sites, I get Pearson R of 0.95 and really pretty good agreement in the quants overall. So yeah, give PTM probing a shot, I would say. And yeah. Okay, future directions, well, ignore this other stuff. I did some AI stuff and I did some graphical network stuff. But importantly, I have a unified, organized database for all these PTMs across the mouse. And really, I could not have done it without having really good organized data coming out of the software. If I had to do this from other softwares, I'd be like pulling my hair out. But because Spectronaut exposes everything you could possibly want and more, it's really easy to make a unified database for all of the mouse PTM data that I have. And that's it. Thank you to the group, and thank you for letting me speak.